For the first experiment, we will observe the catalyst activity. But before we proceed to the experiment, let us first know what a catalyst is. A catalyst is an enzyme which uses hydrogen peroxide, a non-radical Ros, as its substrate. This enzyme is responsible for neutralization through decomposition of hydrogen peroxide by breaking it down into water and oxygen gas, thereby maintaining an optimum level of the molecule in the cell, which is also essential for cellular signaling processes. Now, let's get into the experiment. These are the following materials needed. Hydrogen peroxide, 2 identical shot glasses, potato, ruler, and a syringe. Experimentation proper. First, mark 2 identical shot glasses 1 and 2, and to each shot glass, add 5 ml of hydrogen peroxide. Next, prepare 6 2 cm x 0.5 cm strips of freshly peeled potato tuber. Then boil 3 potato strips in 20 ml distilled water for 5 minutes. After that, simultaneously place the 3 fresh potato strips into shot glass number 1, and the 3 boiled potato strips into the shot glass number 2. Lastly, observe for any gas evolution. Now, let's discuss what happened in the experiment. You should have noticed that the boiled potato strips produced no bubbles. This is because the heat degraded the catalyst enzyme, making it incapable of processing the hydrogen peroxide. While on the other hand, the fresh potato strips produced some bubbles. And this bubbling reaction you see is the metabolic process of decomposition, described earlier. This reaction is caused by catalase, an enzyme within the potato. You are observing catalase breaking hydrogen peroxide into oxygen and water. Catalase acts as the catalyzing enzyme in the decomposition of hydrogen peroxide. Nearly all living things possess catalase, including us. This enzyme, like many others, aids in the decomposition of one substance into another. Catalase decomposes, or breaks down, hydrogen peroxide into water and oxygen. In this experiment, we will observe the bromelin enzyme and the effect of heating on bromelin. But first, let's take an overview about bromelin enzyme. Bromelin contains proteolytic enzymes called cysteine proteinases, which break down proteins and help digest food. It belongs to a group of protein-digesting enzymes that attack the internal peptide bonds of the protein chain. Now, let's move on to the materials needed for this experiment. These are the fresh apple, orange, pineapple, gelatin, microwave oven, and container for the gelatin. Experimentation proper. First, prepare the gelatin. Next, cut 2 equal size 1 inch by 1 inch pieces of fresh apple, orange, and pineapple. Heat a piece of apple, orange, and pineapple in an oven for 1 to 2 minutes. Cool the pieces of fruits to room temperature by submerging them briefly in cold water. Place the regular and heated pieces of fruit on top of the gelatin. Let the fruit pieces sit undisturbed for at least 1 hour. After an hour, remove each piece of fruit and look at the surface of the gelatin underneath.
One hour later.
In fresh orange, the fruit leaves a mark of its shape after it was placed on the surface of the gelatin. The fruit glides indicating that the gelatin became watery on the surface, but the gelatin is still firm. On the other hand, the heated orange leaves a mark of its shape after it was placed on the top of the gelatin. The fruit doesn't glide indicating that the gelatin stays firm. In fresh pineapple, the gelatin liquefies and became squishy. The surface of underneath the pineapple became watery and the jelly setting stops. Meanwhile, in heated pineapple, the gelatin stays solid firm gel, and there is a little amount of liquid on the top of the gelatin. In fresh apple, the fruit leaves a mark of its shape at the top of the gelatin. There is also a little amount of liquid at the top of the gelatin, but the gelatin stays firm. The heated apple also leaves a mark of its shape at the top of the gelatin. The gelatin is solid firm gel, and the gelatin is dry. Discussion of the results. Pineapple contains bromelin enzyme that breaks up the protein in the gelatin. As shown in the experiment, the fresh pineapple melted the gelatin and stopped its setting, making it watery and squishy. Meanwhile, the gelatin in the heated pineapple was still solid gel and firm. This observation is due to the inactivated enzyme. The bromelin enzyme loses its ability to initiate proteolysis, or breakdown, of the peptide bonds that are present in the protein chain of the gelatin. The optimum temperature for activity of the enzyme was 50 degrees centigrade, so when a pineapple was heated in the toaster or oven with about 146 degrees centigrade for 2 minutes, the enzyme present in the pineapple was deactivated. Hence, the enzyme activity of pineapple increased with increasing temperature until it reached 50 degrees centigrade, where it began to decline rapidly. On the other hand, the enzyme phenolase is present in apple, and ascorbic acid oxidase is present in orange. Furthermore, apples and oranges do not contain proteases. For the third experiment, we will be observing the effect of pH on enzyme activity, starch and amylase. First, let's define starch and amylase. Starch is a complex carbohydrate that exists in many foods, including grains, vegetables, and fruits. Starch is a natural polymer, or polysaccharide, meaning that it is a long chain comprising one type of molecule. Starch consists of glucose molecules. It can occur in two forms: amylase and amylopectin. Amylase, on the other hand, can be found in our saliva. It catalyzes the hydrolysis of starch into smaller carbohydrate molecules such as maltose. Three categories of amylases, denoted alpha, beta, and gamma, differ in the way they attack the bonds of the starch molecules. Now, let's get into the experiment. First, these are the following materials needed in this experiment: 5 cups, water, sugar, starch, vinegar, measuring cup, dropper, and iodine solution. Prepare the 5 cups by labeling it as per instructions in the experiment, then add 10 ml of water in each cup. Add the saliva in the cups labeled as saliva, then add 10 drops of vinegar to the cup labeled with acid. Next, add 2 pinches of sugar to the cup labeled with sugar, and 2 pinches of starch to the cups labeled with starch. Wait for 25 minutes. After 25 minutes, add 3 drops of the betadine and observe what will happen. Now, let's discuss what happened to the experiment. The samples change in color because the iodine reacts to a specific substance called starch. The iodine solution determines the presence of the starch in each cup. The first 2 cups became orange-brown in color. This happens because the iodine solution is normally orange-brown in color, and there is an absence of starch in the cups. The 3 other cups turned into purple-black color. The iodine solution detects the presence of starch in these cups, and that is the reason why the color changes. Normally, in the sample saliva plus starch, the correct color for it is orange-brown, because the amylase that is present in the saliva will break down the starch, but it did not happen to the experiment. As what we have discussed in the first experiment, a catalyst is an enzyme that brings about the reaction by which hydrogen peroxide is decomposed to water and oxygen. Now for the last experiment, we will observe the effect of concentration on enzyme activity through hydrogen peroxide and catalysts. These are the materials needed: 6 cups, yeast, water, hydrogen peroxide, syringe, and dropper. Experimentation proper. Put 4 pinches of yeast into 26 ml of water. This will be the solution A. Next, put 1 ml of solution A into 24 ml of water. This will be solution B. In the third cup, put 5 ml of solution A into 20 ml of water. This will be solution C. After setting up the solutions, cover the cups. Now, using a syringe, prepare 3 uncovered cups labeled A, B, and C, with about 15 milliliters of hydrogen peroxide each. Using the 3 covered cups labeled A, B, and C, quickly add 15 drops of each solution to the matching cups. After adding 15 drops each, observe the reaction for a few minutes. As we have seen from the video experiment, less gas evolved as the concentration decreased from cup A to cup C, and that the overall volume of gas also became successively lower in each decreased concentration. This is because there were more molecules of hydrogen peroxide in the higher concentrations, meaning more collisions took place, and there was a greater probability of successful collisions. This resulted in more enzyme substrate complexes formed in the higher concentrations, and less in each decreased concentration. Catalyst dramatically reduces the activation energy needed for the reaction. Without catalyst, the decomposition would take much longer, and would not be fast enough to sustain human life. Hydrogen peroxide is also a dangerous, very potent byproduct of metabolism, and it is essential that it is broken down quickly, so it does not cause damage to cells.